Kirby Bauer disk diffusion method. This disk diffusion method is a gold standard method we are using for confirmation or testing the susceptibility of bacteria. And this standard disk diffusion method was introduced by Bauer and Kirby in 1956. So this is called as disk diffusion method. Why it is called as disk diffusion method? From the disk, and this disk is coated with non-concentration of antibiotics, and from the disk the antibiotics will diffuse into the medium. You can say this is the antibiotic disk, and the antibiotic will diffuse in the medium, and it will kill the bacteria which is near to the disk. So we will get a zone like this. So these are the clear zone, and by seeing this zone, we will get an idea whether the organism is susceptible or resistant to this particular drug. Mullahinda agar or MHA. This MHA or Mullahinda agar is used for this antibiotic susceptibility testing in the case of this diffusion method. And this Mullahinda agar is a non-selective or non-differential medium. Because of this non-selective and non-differential medium, most of the organism will grow well in this medium. And also additionally, this Muller-Hinder agar will contain starch, and the starch will absorb the toxin released by the bacterium, so that the bacterial toxin will not interfere the antibiotics. For this diffusion method, we need a test organism, Muller-Hinder agar, cotton swab, 0.5 McFarland standard, antibiotic disk, and measuring scale. Preparation of inoculum from primary culture plate. So this is the primary culture plate. Here you can see there are so many colonies are there in the primary culture plate, and each colony represents single type of organism. So one colony we will take with the help of this inoculating loop, and we will transfer or we will inoculate this single colony into a liquid culture or broth, and this broth may be sterile water, nutrient broth, or peptone water. And this is the first step. And after that, the inoculum we will compare with this 0.5 McFarland standard. So this is our inoculum, and this is the McFarland standard. And this McFarland standard is a reference standard to check the turbidity or to compare the turbidity of the inoculum, so that our inoculum will be standardized by comparing this McFarland standard with our inoculum. And after that, we will swab the inoculum into the culture medium. This is our inoculum. With the help of a sterile swab, we will take the, we will just dip the sterile swab into the inoculum, and we will just lawn culture the swab to this Muller-Hinder agar. Lawn culture means uniformly we will spread the inoculum to this Muller-Hinder agar. Next step is the application of disc to the media. So this is the antibiotic disc. Antibiotic disc, and this is the antibiotic disc. And this antibiotic disc is a small circular 6 mm diameter filter paper soaked with antibiotic or impregnated with antibiotic. That means simply we can say that. And this 6 mm filter paper will contain antibiotic, and this we will use for antibiotic susceptibility testing. So in this application of disc to this media, we will take the disc. That means depending upon the organism, the selection of disc will be or antibiotics will be different. And this disc, we already told that is a filter paper disc impregnated or coated or added with this antibiotic with non-concentration. Just we will place the antibiotics discs to this lawn cultured Muller-Henner agar medium. Manually we can dispense or we have a dispensaries that is done pre-prepared dispenser is there. With the help of dispenser, just we will place the antibiotics disc into Muller-Henner agar. So after the disc suspension is over, so this is the disc suspended medium. We will incubate this medium at 37 degrees Celsius for 16 to 18 hours or overnight incubation. And after the overnight incubation, we will get an observation like this. So after incubation, this will be our observation. Here you can see some clear area or zone we can see here around the disk. So these are the clear area we can see here. And this clear area means the bacterial growth is not seen in this area, and all the other places or area the bacterial growth can be seen. So after the incubation, we will get an observation like this. And these are the antibiotic discs you can see here. And in some area, we can see that there is no zone. The bacteria is grown all over the area. And in some area, you can see the zone of inhibition. Here you can see these are the zone of inhibition. And this zone of inhibition we can detect by simply seeing the clear zone around the disc. And this is a disc diffusion method. In this diffusion method, what will happen? We already told that this disc will contain antibiotics. If the bacteria is susceptible to this antibiotics, what will happen? From the disc, the antibiotics will diffuse into the medium. You can see the antibiotic will diffuse into the medium. Due to the diffusion of these antibiotics around the antibiotic disc, the bacterium will. So here you can see this is the bigger zone of incubation. That means the diffusion is more here, and this bacteria is susceptible to this antibiotic disc. And here you can see there is no zone of inhibition. That means this bacteria is resistant to this particular drugs. Resistant means during incubation, and this will contain antibiotic. This antibiotics will diffuse into the media, but these drugs will not kill the bacteria. That is why we call it as resistant. Measuring of diameter of this zone of incubation. Here you can see with the help of scale, we will measure the zone of inhibition, and we will note down that zone of inhibition. The measured zone of inhibition we will compare with this standard chart of interpretation. This is the standard chart. This is the antibiotic drugs. This is about the disc strength. This is the diameter of zone of inhibition, that is resistant, intermediate, or sensitive. For example, if you are taking the antibiotic as amikacin, that means we are testing the organism with amikacin, and if you are getting the zone of interpretation or the zone diameter is less than this 14, we will call it as that particular organism is resistant to amikacin. And if we are getting the zone measurement is 15 to 16, in between 15 to 16, we will call it as that particular organism is intermediate to amikacin. If you are getting the zone of diameter greater than 17, we will conclude that the particular organism is sensitive to this humic acid. That means whatever zone diameter we are getting, that we will compare with the standard chart of interpretation. And we will conclude and we will give the result as whether the organism is resistant, intermediate, or sensitive to that particular organism, to the particular drug. Interpretation of zone, the zone can be interpreted whether it's sensitive, moderate or intermediate, or resistant. The sensitive means the diameter will be equal or greater than the standard. The diameter what we measured will be equal or greater than standard, or we will call it as sensitive. And the diameter is moderate or intermediate, we can say that the result is, or our interpretation is intermediate. The resistance means there is no zone of inhibition or less than the moderate, we will call it as resistant. In Kirby Bauer disc diffusion method, first we will take the Muller-Hinder agar plate. To this Muller-Hinder agar plate, we will lawn culture the test organism. And after lawn culture the test organism, we will apply or we will add the antibiotic disc. Depending upon the organism, the selection of the antibiotic disc will vary. And after adding this antibiotic disc to this Muller-Hinder agar, we will incubate the plate at 37 degrees Celsius for 16 to 18 hours or overnight incubation. And after that, we will get the result like this or observation like this. That means in some discs we can see the more zone of inhibition, or in some discs we can see there is no or less zone of inhibition. And we will measure the zone of inhibition by the help of scale. And after that, we will compare the zone of inhibition with the standard chart. And we will interpret the result whether the organism is resistant or sensitive or intermediate to that particular drug.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.